All right, my name is Mackenzie Jahn.
And my name is Katie Richter, and we studied how exposure to selenium affects the expression of the abdA gene and mechanosensation response in Drosophila melanogaster. We chose to use Drosophila as our model organism because of their ease of culturing, low cost of culturing, and they have a lot of genetic similarities to humans, so a lot of the studies done on Drosophila can be applied to humans. We chose to do selenium because it's becoming a lot more of an issue today because it comes from agricultural runoff and runoff from coal-fired power plants and just coal mining in general. And the big deal about selenium is that it ends up in our waterways, so it primarily affects organisms such as fish and birds, primarily vertebrate aquatic organisms. And we chose to do selenium because we're both interested in the environment, and this was something that we both cared about and wanted to look more into. We chose to do the abdA gene because this gene is specifically involved in body segmentation and early development, and then body segmentation therefore leads to the development of a lot of movement-related structures such as the spine, limbs, fins, and tails of aquatic organisms and aquatic birds. And we do notice that in the research, it shows that a lot of these animals that are affected by selenium exposure have spinal deformities, like the one you can see in figure 1 over here. And then to select our amount of selenium, we did some research to find what was considered lethal or what was considered unfit for human exposure. And the deadly level of selenium is thought to be about 2 micrograms per liter. And so we chose to do 1.5 micrograms per liter, which should be enough to produce a negative effect on the larvae, but not enough to kill all of them off, because then we wouldn't be able to study their movement effects. And so our hypothesis was that exposure to 1.5 micrograms per liter of selenium will cause expression of the abdA gene to decrease, and then that will therefore cause our mechanosensation response to decrease as well due to the impairment of movement-related structures. And now Mackenzie will go over our methods.
All right, so in order to do this, we had to create both control cultures, controlled Drosophila cultures, as well as experimental Drosophila cultures. So we did this for the control by adding our potato flakes, which is the growth media for both the control and experimental. And with the control, we added DI water in order to hydrate those. And for experimental cultures, we created a solution of 1.5 micrograms per liter of selenium and hydrated it with that. And because 1.5 micrograms per liter is a very small concentration, we created a more concentrated version of that and then did 3 serial dilutions to then get our stock solution, which we used. And we created these cultures 1 week before we ended up using them, so they had time for larvae to develop and be created. And for our mechanosensation assay, as I said, we did all assays 1 week post creating the cultures. And so for our mechanosensation assay, we did this on 3rd instar larvae, which are the biggest larvae, the most developed larvae, and they're sitting on the top of the growth media. And so we did this by taking a larval scoop and then washing the larvae with the PBS solution to get the growth media off of them. And then once we did that, we took a dissection probe and we gently rubbed them along their sides. And if there was a reaction, it was fairly obvious and we would mark that. And if there was not a reaction, we would mark that there was not. And we did this on 3 controls and 3 experimental cultures, and then performed statistical analysis. And then for doing the RT-qPCR, we first had to do RNA extractions. So we extracted RNA from also 3rd instar larvae, which we washed in PBS just as before. And we extracted the RNA using the RNeasy RNA extraction kit. And after we extracted RNA, we were able to perform reverse transcription to create cDNA, which we then ran qPCR on. And we ran qPCR looking at the gapdH gene as well as the abdA gene. We looked at the gapDH gene as a control just to make sure that qPCR was actually running correctly. And as well as doing the RT-qPCR and the mechanosensation assay, we ended up doing a larval count as well, which in order to do the larval count, we took the top 1/3 of the media and we washed it in PBS just as before, and we counted the third instar larva. We ended up doing this larval count, which we hadn't previously planned on doing, because due to the coronavirus, we were actually only able to run 1 round of RT-qPCR as opposed to the mechanosensation assay, which we were able to do 3 of. And because we couldn't perform any statistical analysis on 1 round of PCR, we decided to do a larval count as well, which is why we didn't have anything in our hypothesis about that. So anyways, with the larval count, as I said, we just did the top 1/3 of growth media because that's where most of the third instar larvae reside anyways. And if we took all of the media, we would be counting for days. So we just did that. And then Katie will talk more about our results.
Alrighty, so if you direct your attention towards figure 1, this is an image from Frederick Freyer of the USGS in California, and he studies the effect of selenium on aquatic organisms. And this is one example of what can happen to fish, and spinal deformities are actually the most common effect of selenium in fish. And this is a young, probably 1 or 2 year old minnow from the Sacramento splittail species, and this is concerning because they are the last fish of their genus. All of the other ones have gone extinct due to overfishing, so it's really important that we keep this species alive and well, but with exposure to selenium, it could become a little bit difficult to do that. And then looking over at figure 2, this graph shows our fold change in expression of the abdA gene. And as Mackenzie mentioned, unfortunately, we were only able to perform 1 round of qPCR, so we don't have any statistical analysis on this. But if you look at the bar graph, you can see that the control level of abdA is relatively high, right at 1, and then the selenium treatment is way down there at just below 0.2. So that shows a very substantial decrease in gene expression, just as we predicted. And we also believe that if we were able to perform statistical analysis by doing more rounds of PCR, that this would yield some statistically significant results just by looking at the vast fold change there from our qPCR. And then down in figure 3 is the results of our mechanosensation assay. And if you look on the y-axis there on the left, the numbers go from 0 to 0.9, and that's because in order to perform the analysis on this, a response, so as if we poked the larvae and it moved away or responded somehow, that was recorded as the number 1, and no response was recorded as the number 0. So our control is all the way up there at about 0.75, and the selenium is about at 0.5. And if you take a look at our p-value down there at 0.0046, this shows that the difference between the control response to mechanosensation and the selenium treatment response was significant. So we can conclude that this difference was not due to random chance, and the differences in mechanosensation were likely due to exposure to selenium, which is really important to note because there isn't a vast amount of research out there at this moment about how selenium affects aquatic organisms aside from the actual physical deformities. And now we know that it does affect movement ability in very young Drosophila larvae, so that might be something to look into farther. And then finally in figure 4, we have our larval count bar graph. As you can see, the control amount had a little bit more larvae than the selenium treatment, but if you take a look at that p-value of 0.55, that was not a statistically different level, although that's not necessarily unexpected because we do know that selenium, at least at this exposure level, does not necessarily cause mortality in larval stages or in very early development. It just causes deformities, so we really would not expect to see a significantly different amount of larvae in either culture. And then Mackenzie will go over our conclusions.
All right, so as Katie mentioned, our behavioral assay showed a significant decrease in mechanosensation for the third instar larva of the selenium exposed group compared to the control, which as shown is in figure 2. And that again basically states that there is a significant difference between how much the larva will move based on the selenium exposure, so that's pretty exciting to see that there was significance there. Again, because Katie said there hasn't been a lot of research on that. And the results for our RTQ-PCR showed a decrease in the abdA gene expression for the selenium exposed larva, which is exactly what we hypothesized. However, because of only being able to do one round of qPCR, we can't determine any significance. But as Katie mentioned, we do believe, based on the substantial difference that we can see on that bar graph, we do believe that if we were able to perform more rounds of this, that we would find significance. And we also, as mentioned, looking at figure 4, there was no difference in third instar larva numbers between the selenium treatment and the control group. And we would like to mention that some sources of error could be that our larval count, as we stated earlier, we were just looking at the top 1/3 of media, which was basically going on, this looks like 1/3. And so that could be a source of error. Also, another source of error with that is that the larvae do swim around because they are alive. So they're swimming around in that PBS solution when you're counting them, making it kind of difficult to count. So that could be a source of error. And another possible source of error is that although we found that the mechanosensation response was pretty obvious, that is still based on our eyes. It's not like running PCR where it's based on pure data. Mechanosensation is a behavioral assay that we are saying, yes, this happens, or no, it's not. So although we believe we did a good job, there's always a possible error there. And for future experiments, really we'd just love to be able to run more rounds of PCR. We think that if we were able to run more rounds of qPCR, we'd be able to find some significance. So we would just like to do more rounds of that, as well as, given that we found some interesting information on the mechanosensation response, we'd like to do more rounds of that as well, just to see if we'd continue to find that and yeah, 2 more rounds, possibly change the amount of exposure to see if that would change it even more.
Thank you.